Well, hi, everyone. Really excited about our call today. And, a lot of the last, is this 5 months now? We've been doing as many Monday calls as we can. And really, they've been related around the markets and what's been going on with the markets in reaction to COVID. But today we have something very different, and very excited to have the La Jolla Institute of Immunology on the call today. And they're also in our backyard, which is great. So we have a really great resource here for the call today. And we've also been gathering questions that all of you have provided to us, but we're more than like to take more questions during the call. If you have, there's a Q&A that you can text into the text box, and we'll try to get to all those questions. So thanks again for everyone for being here, and also to the group here of coordinating all of this. So we're really excited to have Dr. Saphire here today. I want to read a little bit of a bio on her because I think it's really important to understand the level of expertise that we're going to hear from today, and also really the impact that the La Jolla Institute of Immunology is also providing for this COVID. So give me a minute just to read from this so that we can, again, have a little bit of depth to Dr. Saphire. So she is a professor at La Jolla Institute for Immunology and a director of CoVIC, a Gates Foundation-funded effort to understand and deliver antibody therapeutics against SARS-CoV-2 to ensure that no one is priced out of a life-saving therapy. Her research combines structural biology with cellular virology and immunology, and has revealed the structures of the trimeric, gosh, am I pronouncing this right, perfusion glycoproteins for the Ebola, Lassa, Sudan, and Marburg viruses, neutralizing and non-neutralizing antibody epitomes. I'm blurring that word. How these viruses drive themselves into cells and how they suppress immune function, and where the viruses are vulnerable. Her work has begun recognized at the White House with a Presidential Early Career Award by the Burroughs Wellcome Fund, by Young Investigator Awards from the AASB, ASM, and the International Society for Antiviral Research, and by the Saran Sidhu, pronounce that right, hopefully, award for the most outstanding contribution to the field of the diffraction. She is a fellow of the AAM and the AAAS, and serves on several scientific leadership boards of institute nonprofits for medical research for human health. So we have such a phenomenal resource here at our fingertips to provide us not only what is going on with COVID, but where they see things going today, tomorrow, and 20 years from now, 5 years from now. So we're really excited to have Dr. Saphire here. I'd like to actually hand over the mic to have Mr. Lee give us a little bit of an update of what La Jolla Institute for Immunology is and what they do. So if I can hand that over to you, that'd be great. Thank you again for joining us.
Yep, such a pleasure to be here. Good afternoon, everyone. After that great intro to my expert that's coming next, I have to be on the screen to bore you for a moment or two, but you're really going to be, your socks are going to be knocked off from what you're going to hear from Erica and her direct work in this very important subject. But I wanted to just give a little context to who we are. If you all were here with me in person, I'd ask you to raise your hand who knows of the La Jolla Institute for Immunology. And then usually I ask who's been to the La Jolla Institute for Immunology. That used to be a lot easier before COVID, obviously. And we want you to know that once COVID ends or life gets somewhat back to normal, we'd love to have you all out to visit the Institute. It's a real gem, not just for the area, but for the world. And too often people in San Diego, our own backyard, don't know who we are and why we are so significant. So just a couple little facts. We've been around almost 32 years now. We have been one of the world's most significant organizations focusing on the immune system from the very beginning. We have about 500 people who currently work at the Institute. Most of those are people who hold either a PhD, an MD, or both. I'm one of the rare exceptions who doesn't. but I'm really happy every day to be able to work with such talented people. We're a larger organization in the context of, again, without people not knowing much about us, in the sense we're about a $65 million a year organization, and that will be growing with the success you'll hear about from Erica and others at the Institute in addressing the coronavirus pandemic, or the COVID-19 pandemic. Most of our funding is through the NIH, which basically speaks to the excellence of our research. Those are very highly sought after, hard to get grants that only the best science receives, and a significant portion of our work has always been funded by that very important source. We have also a number of business development opportunities where some of our ideas and technologies are going to market, which is allowing us to get, again, our great work into patients, but also is leading to some revenue back into the Institute and back into the science. But lastly, we are really relying, in this case, quite heavily on private philanthropy, not to do the long-term work that Erica is an expert in, and most of our researchers are very skilled in achieving these large-scale grants, but to get things moving quickly. And as we know, in this world of COVID-19, quick is important, and for all of us, it almost seems like it's not quick enough. But the only way we're able to really launch a lot of these initiatives quickly is through private philanthropy. Quickly, just two more topics, going to the next slide. You know, again, before COVID-19, maybe not a lot of people had really understood the power of the immune system, and certainly the power of studying the immune system. And now it seems to be everyone's part-time hobby is understanding the immune system and the terminology and the work that goes into that. You all are lucky. I feel lucky that on a day-to-day basis, we have some of the world's most significant experts understanding how the immune system works and how that could turn into various technologies and results that could contribute to the betterment of human health. Next slide. And as an example, of course, we've been heavily involved in the COVID-19 work. Last year, we launched 3 focused centers in our work to address areas of, a range of human health conditions that all have beginnings in understanding how the immune response or the immune system can help us, or in some cases harm us. So understanding it is very important for any number of conditions. So our 3 centers we had launched at that time were a Center for the Study of Autoimmunity and Inflammation, the Center for Cancer Immunotherapy. We have a current cancer immunotherapy treatment in FDA trials, for example, showing the promise of that area of work. And then in the infectious disease space and vaccine research space, we clearly look like visionaries by launching this center when we've become one of the national leaders in this space, and Erica certainly is in that category. So again, something that I hope San Diego and all of you can be proud of what's happening in your backyard. Next quick slide. When the Institute first was aware of the COVID-19, please move to the next slide if you can. We launched a task force, if you will, Erica front and center in that work. But a number of our principal investigators, our top faculty, have directed lots of their effort, which was already incredibly taxed with other important studies and work, to really address some of the understandings of COVID-19 and how we could then move forward with some potential solutions. So you probably have seen a lot of these people recently mentioned in the news. We've been all over the news here in San Diego, nationally. New York Times, for example, just featured some of our work, and even internationally. So again, this group is not only a task force, but frankly, a group of superheroes. To the point, next slide, another sort of, sort of a point of pride for the Institute, but certainly shows the importance of our work. Dr. Fauci spoke a few weeks ago to Congress on all the COVID-19 efforts that are underway, particularly those funded by the federal government. And of all the work he referenced, he held up a paper by two of our top faculty members on their work, speaking to the significance of what we do. And in Dr. Fauci's case this day, speaking to the significance of what the research community is doing in order to better understand this condition so that we could hopefully get to a normal world soon enough, or a new normal world soon enough. But it was a very important moment for the Institute, and it again spoke to the power and the significance of our work. So without further ado, I'd like to just turn it back over to the person who is day-to-day, and you'll see her in her lab, making great strides and progress in understanding this virus and helping us all, again, get through all of this and move on to happier, brighter, and more healthy days. So I'm pleased to introduce Dr. Saphire, who is, as you heard earlier, an incredible expert and a great colleague. And I'm sure you'll enjoy the presentation. Thank you.
Thanks, Chris. And thanks everyone for joining us today. So I'm very happy to be with you here from my lab at Jolla Institute. And I'll show you what we are doing with our new research, what we are understanding, and where the nation can go from here. So let me share my screen. There, can you see that? As you all know, we are all at home and we are waiting, praying for immunity. Immunity in the form of a vaccine, or immunity in the form of immunotherapeutic. We are 6 months into this pandemic, and in 6 months, we've had nearly 20 million infections, and it's spread around the globe. It has affected more than health. Our economies, as you well know, are struggling with rising unemployment. Shopping is down, and when we tried to open schools, in some places, they closed right back up again. And so we need that immunity in order to go back to normal and return to life as we wish to live it. The trouble is that getting herd immunity from natural infection is too slow and too painful. So I think that probably about that 20 million cases that we've noticed so far represents about 2.5% of the world's population. So 97.5% to go. Even if we are underestimating the number of cases, achieving herd immunity by natural infection is going to take too long and result in too much loss. We need vaccines, and we need them faster than science has ever discovered or mobilized them before. Now, the process of discovering a novel vaccine typically takes years, 10 years. The fastest we've ever done was mumps in 4 years. This time, we're compressing it from years to months. Now, a vaccine pipeline begins with preclinical studies, then proceeding to phase 1, phase 2, and phase 3 clinical trials. Preclinical is where I work, and that is where we do a lot of different things to decide which vaccine goes on to the path. We make revisions when things don't look like we hoped in those phase 1, 2, and 3 clinical trials. We do a lot of work to understand the pathogen. More importantly, we like to understand where that pathogen is vulnerable to immune attack. We look at the people who survive and the people who die, and we try to understand the differences. We look, therefore, at what we call the correlates of protection. What is it about the immune response when someone's been asymptomatic or they survived that let them survive, and how do we inspire exactly that, but better and longer lasting in a vaccine? How do we mimic the virus to show the right shape, right structure, right molecule to the immune system to have that immune response primed and ready to go before we get infected? And we test that to see if we're getting the right responses that we think we have. We work in cell culture, we work in animal models. And we try to understand if the vaccine has elicited 3 kinds of immunity: antibodies, T cells, and innate immune responses. When we found good candidates, and this is often a cyclical process going back and forth of preclinical, we enter phase 1 trials. Now, phase 1 trials are typically a small number of people. They are healthy, immunocompetent adults. And we look at a couple of things. Is the candidate vaccine safe? Does it give them some sort of unfortunate or difficult side effect? Does the vaccine elicit the immune response we were hoping for? Have we picked the right dose? Do we need more? Do we need less? After testing the waters in a few healthy people, and it might be groups of 15 to 50 people, we launch a phase 2 trial. That's bigger. That could be hundreds or thousands of people. And that aims to develop on more information on a deeper level. Does a vaccine work better in different ages? How about ethnicities? How about genders? If you had pre-existing antibody or pre-existing immunity from this virus or something similar, how does it affect your response to the vaccine? Have we picked the right dose? Have we picked the right schedule? Meaning, should we give 2 doses? Do they need to be 1 month apart? How about 6 months apart? And what is the outcome? Are these people protected? How long does it last? What immune responses specifically did the people that fought off the virus make? And we can use that to inform phase 3 trials. Phase 3 are the final arbiter. Phase 3 involves tens of thousands of people, and it tests the final vaccine formulation for 2 things, safety and efficacy. Is it safe? And does it work? And then we look on that population scale of tens of thousands of people to see, did it protect the vaccine recipient? And also, did it protect the recipient's contacts? Because the goal is to not just keep the person who received the vaccine alive, but keep them, if they do become infected, from spreading the virus further. The more people we vaccinate, the more we can protect the people that they live with and the people that they work with. Now, right now, there are 165 vaccines against SARS coronavirus 2 in preclinical studies. I have one in my lab too, so that number is 166. Well, probably more than 166. Of those, there are 29 that are currently undergoing human clinical trials. There are 18 phase 1 clinical trials going on around the world, 12 phase 2, and 7 phase 3. Now, if you're adding together those numbers, 18 plus 12 plus 7, that's more than 29. And the reason is that some vaccines launch more than one phase 2 trial. They might launch it in different places, maybe in the USA and maybe also in Brazil, or they might look at different formulations in different ages and different uses. Now, important thing to note is that we are fairly desperate for a vaccine against COVID-19. And we, as a nation, will consider that candidate vaccine effective if it protects half of the recipients. You would not consider your measles virus vaccine effective if it only protected half. But we're aiming for something as opposed to nothing in this accelerated timescale. And it's very likely that the first vaccines out won't be the best vaccines, that we'll have a chance to refine and improve because we're expecting seasonal return of this coronavirus. So maybe more in development. Now, that's the pipeline. How does the vaccine actually work? So the coronavirus has a protein on its surface that you've heard about. It's called the spike. It is what gives the corona, the crown on coronavirus. That spike protein is what the virus has and uses to attach to your cell and drive itself in. That is the machine the virus uses to infect. That is also the target of your immune response, especially for antibodies. Antibodies are Y-shaped structures that anchor onto that spike and inactivate it. So that antibody, whether you get it after your natural infection or you've inspired it with your vaccine, has 2 major jobs. The first job is to bind onto and inactivate the virus. We call that neutralization. And that works like a club on the steering wheel of a car. It just prevents that machinery from working. You can't drive the car, or a boot on the tire. It's dead in place. That spike isn't working anymore. The second job, an equally important job with that antibody, is to recruit the immune system to help. Not just to stop that virus, but to alert the immune system of the presence of an infection so that immune cells can come and clear the virus from the body or clear infected cells so they stop being virus factories. So antibody has 2 different jobs and they're both very important. You might also want a vaccine to inspire T cell help. And T cells come in different types. We have some kind of T cells that are killer T cells, and those are the ones that antibodies inspire responses from to come in and destroy infected cells. There are other T cells, helper T cells, and those are what cause you to make more of the immune molecules you need to fight an infection. There are different kinds of T cells. You want them both. And these antibody and T cell responses talk back and forth to each other. You need the different kinds of cells operating simultaneously in order to mount and maintain that immune response. You know, the polio vaccine you got, you know, the measles vaccine you got gave you lifelong immunity. We want to be able to inspire memory here as well, and that's going to take some work. So that spike protein is key. And that spike protein is the major component of all of those vaccines that are under development and in clinical trials. But they each display that spike protein in a different way. So there are different types of vaccines. There were vaccines in which you take that actual virus, that actual SARS-CoV-2, and you kill it or you weaken it, and you show the immune system a killed or a weakened virus. Another route of vaccine is to just take the genetic information that encodes the spike, not the spike itself, and you deliver that genetic information, the gene, could be RNA, could be DNA, in a package with larger molecules like lipids or other things. That's genetic immunization. A third mechanism of immunization is to take the gene that encodes that spike protein and you deliver it not in a ball, but on a carrier virus, some kind of little harmless virus, something that really only infects animals or that will infect your cells but not cause any disease. And sometimes that viral vector just delivers the payload of the gene. Its job is to just get that gene into your cells so that you can show it to your immune system in time. And sometimes that viral vector displays the spike protein on its surface, like the blue one on the right. And the fourth type of vaccine is just that purified spike protein itself. And sometimes they aggregate them together and display them in kind of a ball. Now, there are 3 vaccines in clinical trials that involve inactivated or weakened actual SARS coronavirus 2, and all 3 of those are mobilized by Chinese companies. Those are not the ones being forwarded in the United States because for novel vaccines, it can be difficult to understand if you have really inactivated or weakened that virus very well. What the United States has invested in with Operation Warp Speed and other efforts are these types of vaccines. And they're investing in many of them with the idea that you take a lot of shots on goal, one is more likely to succeed. Moderna and Pfizer have made RNA vaccines, where Moderna is injecting RNA that encodes the spike protein. It's a type of RNA called mRNA. It's the one that actually delivers the message to make the spikes. Your cells will start making that spike protein. Your immune system says, I don't like that, that's foreign, and has an immune response mounted against it before you get infected. Pfizer has 2 vaccines. One is the entire spike, one is just a piece of that spike, also RNA. There are 2 companies that are mobilizing viral vectored spike, AstraZeneca and Janssen, a division of Johnson & Johnson. In AstraZeneca's vaccine, they have the spike gene expressed by a type of virus called an adenovirus. It's a weak virus. It causes a common cold or a little eye infection. And the ones that AstraZeneca is using is a virus that typically infects chimpanzees. It will also infect your cells. Janssen and Johnson & Johnson are mobilizing spike displayed by a human adenovirus. Now, these are weak viruses. You don't want to cause disease with your vaccine, but it often means that they might get weaker responses. And then Novavax is also delivering a vaccine that's made from the spike protein itself. Lots of copies of the spike protein gathered, like I've shown you here, in a snowball or something called a nanoparticle. So those are the different types that are being mobilized. And so the question we want to know is, do each one of these different vaccines give the kind of immune response that we want, a potent one, a protective one? And the other question is, if they give that response, how long will that last? Are we protected, and for how long? So we can take a deep dive into one of the front runners, the Moderna vaccine that Operation Warp Speed has invested heavily in. The phase 1 data, safety and efficacy, came out for the Moderna vaccine a couple weeks ago, 2 weeks ago. And in that phase 1 trial, they looked at 45 healthy, immunocompetent adults. They divided those 45 people into 3 groups of 15. Each one of those groups got a different dose. So 15 people received 25 micrograms of the mRNA. 15 people got 100 micrograms, and 15 people got a 250 microgram dose of the mRNA that expresses a coronavirus spike vaccine. Everybody got 2 shots, and they got the shots about a month apart. So if you were in the first group, you got a shot of 25 micrograms, you come back a month later, you got a second shot of that 25 micrograms. And then they studied those recipients for 2 months. So we know whether or not this immune response lasts 2 months. We haven't had time yet to know if that immune response lasts 2 years or a lifetime. But the first question is, we want this vaccine to inspire an antibody, these Y-shaped molecules. So does it? In the phase 1 data that they published here in the New England Journal of Medicine, after the first shot, the 25 microgram people elicited 40,000 units of antibody. The high dose group, 250 micrograms, 210,000 units of antibody. So you get more antibody with the bigger dose. And after the second shot, you get more antibody yet, where a boost from 40,000 to 300,000 units, or a boost from 200,000 to 1.2 million units. So you do get antibody. You get more with a bigger dose, and you get more on that second shot. You get the right kind of antibody, right? The two jobs of antibody are to anchor to and inactivate or neutralize that spike and inspire immune protection. So antibodies bind and antibodies neutralize. We think the neutralizing ones are what we really want from that vaccine. Unfortunately, the antibodies that resulted after that first shot really don't neutralize. They can detect the spike, but they don't inactivate it. But after the second shot, these people did make antibodies that could neutralize the spike. And the kind and potency of the antibodies they made were as good as immune responses from people that survived. In fact, the top half of that immune response of survivors. So that's good. After that second shot, they make antibody, and that antibody can neutralize the virus. Well, the next question is, when do they make these antibodies? We need protection now and we need it to last. So if you look at the antibody response graphed here by the group that got the low dose, the group that got the medium dose, the group that got the high dose, and you look at the time points. So this is when they got shot number 1 at day 0. Nobody really has antibody. After 15 days, people start to develop antibody against the spike. So it takes 2 weeks to make that first response, and it's a steady rise. But then it's a plateau. See, all those lines level off. They don't start making more antibody until they've gotten that second shot a month later. And you can see another boost and then a plateau at the top. They followed those people for 2 months. So they kept their antibody for 2 months, but they needed 2 shots to get enough and 2 shots to get the type of antibody we want to have. If you look at the high dose people, that's where they began. They have some antibody at 15 days, but even though they got that high dose, they still need that second shot to get the higher levels that they see at 2 months. So they start making some antibody in 2 weeks, but they need that second shot, and they don't really have that full amount of antibody on board until 36 days. So 5 weeks after you've gotten that vaccine, you're not protected for 5 weeks. Okay, so that's when they made any antibody at all. The really important thing, though, is when do they make the good kind of antibody, the neutralizing antibody? And that's an important difference. If you graph the neutralizing antibody, it looks like this. So if you look at the bottom left, the low dose group, that's when they got their first shot. Nobody really made neutralizing antibody except one guy, and then it fell. When they got the second shot, they boosted not just the more antibody, but they proliferated in making the ideal kind of antibody, the neutralizing antibody. The same thing with the high dose group. Even though they got that very high dose of the mRNA spike, it really wasn't a strong neutralizing response, and it sort of fell apart until they got that booster a month later. You gotta have that booster. Now look here also, on the bottom left where I've drawn that blue circle. That is a tremendous range. That is each one of the 15 adults that got that low dose vaccine. Some of them made a good amount of neutralizing antibody, some of them never did. There's a lot of people in the middle. There's a big range in how people respond, especially at that lower dose. If you look at the higher dose, they've all done better. So a bigger dose might be better, but there's still a range there. Some are potent and some are not. There's individual variation. So that's 2 months, but we want to know what's going to happen through the fall, through the winter, next year. Will these people have memory antibody? And the answer is that we don't really know. It is a new virus. This is a new vaccine. This paper came out 2 weeks ago. They were only able to follow these people for 2 months. We won't know for 6 months until they were protected for as long as 6 months. So we will follow that. Now, this is also just healthy people. They look to see, did you make an immune response? These people were not, as far as we know, exposed or what we call challenged with the virus. We don't know if they were protected. They could do an animal study, a primate study, with this same vaccine. And they could show that 1 month after they got that second shot, when the animals were exposed to the virus, they all survived, and immune response that all these animals had was able to limit and control viral replication. So the vaccine after the second shot does protect against the virus at least 1 month later. We won't know until 6 more months from now if it kept that immune memory 6 more months. So that's data that the field still needs. So that was antibody. Did these people make T cells? The answer is, we think so. So they've only reported the T cell data for the low and moderate doses, not yet the high dose. And it looks pretty good. They made the right kind of T cells. They made the helper T cells, and they made it in that right balance. And it looks appropriate for an antiviral response. So that's pretty good news. But the other thing you want to know, not just efficacy, does it work, but is it safe? In a virus for which 80% of cases are asymptomatic, you don't want to induce disease where there wasn't going to be one in the first place. What side effects did the vaccine give? Well, there were reported fatigue, chills, headache, fever, pain at injection site. That's the hallmark of an immune response. When you get fever and chills from a flu vaccine or flu virus, that is evidence that your immune system is creating that antiviral response. So they did get some side effects, but it also means their vaccine was working. But the side effects differed in the dose received and on whether it was the first or second shot. The low dose people, remember these people didn't make as much antibody, and there was that big range. Only a third of them had vaccine side effects. The high dose, half of them did after that first shot. After that second shot, where you have a stronger immune response because you've seen that thing before, half the low dose people had some side effects. Everybody in the medium to high dose had some of those side effects. One of the low dose people ultimately had to drop out of the study because they got some hives and swelling in their legs, I think was related to that first shot. But I think it's important to note that the immune response to this vaccine is significant, and you will feel something, especially on the second shot, and especially with the higher dose. Some of them reported severe fatigue, severe headache, severe pain and other discomfort. But none of these things were life-threatening or a great concern to the researchers. So here's some good news. The vaccine recipients made the kind of immune response that we would hope they would make, and it lasted at least 2 months. And there were no serious adverse effects. But there's also some reality that we need to face here. You're gonna need 2 shots. One's not enough. You won't feel well after that second shot. You might be in bed, and maybe that's worth it to avoid an actual coronavirus infection. The issue for public health is that it can be hard to get people to show up to get that booster shot. And older people will probably need that higher dose that gave more side effects. An older person's immune response is not as robust, and it might, for studies of flu, they needed 4 to 6 times as much material to make the right response. So we might expect a higher dose in older people. The other dose of reality is that even though a vaccine might be approved, it's not going to be a magic bullet. It's not going to be, we were at home and all of a sudden we were all out resuming our lives, because that vaccine is not going to be in all places at all times. The key day is not when the first person can go to that first doctor's office and get the first shot. It's when the population really of the globe is protected, when the vaccine can be everywhere and we have broad coverage of herd immunity. As we have seen this year, if this virus is anywhere, it will rapidly become everywhere. It went from just a handful of cases in Wuhan in the end of December to a global pandemic quite rapidly. Viruses can circle the globe now in less than 24 hours. If there are pockets or countries where the virus propagates, it will come back. So we are not safe until we have broad immunity from vaccines everywhere. The other issue to face is that there is quite a range of responses. Some people will respond well, others will not. Sometimes a vaccine looks like it just doesn't take. And it's going to take time to get there. So you're not protected the day you walk out of the vaccine office, not even the day you walk out of that second vaccine office. It's going to be 5 or 6 weeks after the second shot, or first shot, before you get some protection. And we still need some data. We need to know if these people who made these immune responses, if these were enough to protect them in the real world, not monkeys in the lab, but real people in real exposure. We want to know who it protects and how many it protects. That range of responses, is it linked to immune condition? Is it linked to prior exposure to something? Is it linked to age? Is it linked to ethnicity? We know that the virus is affecting some communities more than others. It could be sociological, meaning they're essential workers and they live in multi-generation households, or there could be some underlying genetic thing in which the immune response to the virus and response to the virus, susceptibility to the virus actually differs. And the other key feature is how long will protection last? Is it lifelong, or will you need to get a booster every so often every year? So these phase 3 trials are now ongoing, and this is the data that we are all waiting for. This will take some time to enroll the tens of thousands of people they need to look at safety and efficacy on a larger scale and wait and see if those tens and thousands of people have gotten infected. So it takes time to enroll them, it takes time to vaccinate them, and then it's going to take some time to wait and see if they are protected as they go about their day and go about their lives and jobs. Were these people protected from disease more than people who received the placebo? What we don't yet know is if older people would be protected. And these are the people that we most want to protect because they are at most risk of getting severe disease. We don't know how long protection will last. We don't know what that phase 3 data will tell us. We don't really know when they're going to be available. The most optimistic estimates are January. Others say later on into the spring of 2021. And we don't know how many doses we can make. Manufacturing capability has been secured and ramped up, but there's an anywhere, everywhere problem. We can vaccinate ourselves here, but will the virus return on a flight from somewhere else? The other things, which are based on our prior experience working with all kinds of other viruses and vaccines, is this. We're aiming for something fast, right? We just need some vaccine now to give some immunity, and we're going to call it effective if it protects 50% of the people. We know that vaccines often do not work as well in older people, and that is tremendously unfair right now because those are the people that we need to protect. We know these vaccines do not work in the immunocompromised, also people who are at greater susceptibility to SARS-CoV-2. The other issues is that not everyone can be vaccinated. Pregnant women, babies under age 1, the immunocompromised, often cannot receive some kinds of vaccines. The other issue is that not everybody will be vaccinated or vaccinated yet. They might be willing but haven't gotten that vaccine yet, or they might refuse it overall. And again, if we don't have herd immunity, we are not safe, even if we ourselves have been vaccinated, if that immunity doesn't last. We can expect global travel and seasonal return of that virus. And even when the vaccine is available, you won't have antibody on board until a month after you receive it. So if you don't want to wait a month and you want protection right now, or if you are among those for whom that vaccine didn't take, those people at the bottom that never mounted an immune response, and you get sick anyway, what can we do? What if you're that guy? Well, there is a way to deliver immunity immediately without needing a vaccine to try to inspire it. And that's called antibody therapy. That's delivering antibody right away as a drug. And antibody therapies are the fastest growing group of biological therapies there are now. We have antibody therapies mobilized against cancers, against infectious diseases, against autoimmune diseases. You, of course, have heard of antivenom. If you get bitten by a snake, you get a shot of antivenom. That's the antibody against the snake toxin. If you get that antibody, it will save your leg and save your life. And with antiviral antibody, just like with antivenom antibody, earlier is better. You can actually make in the laboratory large doses of potent antibody and just give it to somebody IV. So if they're infected, you keep them from getting sick, or if they are a person at high risk of exposure, like a frontline medical worker, you can prevent them from getting sick while we wait for a vaccine or if the vaccine didn't work. Now, the good thing about delivering antibody as a drug is that we really understand how antibody works. Antibody is a natural human molecule. You make it yourself. Antibody against SARS-CoV-2 is what you would have made yourself if you had had the opportunity, if you had a good vaccine, or if you had survived. Because it's a natural human immune molecule, it's a very safe biological format to give, and it's transient. Antibodies last about 3 weeks. If you engineer them for a longer half-life, they'll last about 6 months. So there's no fundamental change in your immune system or immune response. It's just temporary protection that's going to get you through a crisis in a very safe way to prevent you from getting infected or treat the sick. And these are important, and the US government knows it. So Operation Warp Speed is investing in antibody therapy too as a complementary approach for vaccines. Now, these antibodies that we are looking at to figure out which ones should be delivered as drugs are those that have made it by survivors, people that have survived COVID-19 and have a good immune response. But these people make a lot of antibodies. There are a quintillion possible antibodies in human sera. So how many is a quintillion? Well, my father's a math teacher and I am my father's daughter. This is a quintillion. You make this many different kinds of antibodies in your blood right now. Among those, we want to find the very best one or two, sometimes three, probably two, that can be given as a drug. So we need to find among that massive haystack the two needles that are going to be most effective at anchoring to and inactivating COVID-19. How do we find those? Which antibodies of that tremendous array are best? And how do we know they're best? What is it we can do in the lab to figure that out? Well, an antibody is shaped like a Y. It has two arms and has a base. The arms are hypervariable. They have a very specialized binding site. So that's U, that's the foreign invader, the virus. It has, at the end of the arms, fingers that offer something like fingerprint-like recognition that make that antibody specifically targeted to one place and one pathogen. And so we can look at the ones that hit exactly the right spot on the coronavirus and have the right effect. Now I've shown you a Y, this is what it really looks like. In blue is that Y-shaped antibody molecule, and here it is anchoring very, very tightly onto a viral surface spike and activating it. That's what it looks like. Those are the arms that fingerprint recognition. The base of the Y is what recruits immune cell help to destroy that virus and antibody and clear and virus and infected cells from immune system. Now, we need antibody therapy, and we need it 3 months ago. There are at least 60 companies right now that are racing to find the very best needles in that haystack, the very best antibodies, some different survivors that looked at of COVID-19. And each one of these companies is using a different strategy to find them, characterizing different therapeutic candidates. We want to know across this playing field of potential products which ones are best and how we know they're best. Because every company says they have a really good one. How do you know which are the best? How do you choose which of these 60 you want to try in people in these clinical trials? You can't try them all, you have to find the best. How are we going to do that? How are we going to look across this global field of competing companies that each say they've got the best therapy and figure out which really is the best? Because we need to find the best. We need these antibodies to last. We need these antibodies to be useful in the winter and in the spring and next year and the year after that. This is going to be a while before we're done with coronavirus, and it's going to mutate in between. We need an extraordinary level of safety. We need, because antibodies are ideal for people that cannot receive some kinds of vaccines, like pregnant women, older people, the immunocompromised, frontline workers, babies. We need an extraordinary level of safety of these molecules. And we need to work very quickly to find them. It's not going to do us any good if we take 2 years to figure out the best therapy for something that we needed a year and a half ago. And we need to analyze this broad playing field of antibodies being mobilized by small companies, multinational corporations, and academic labs. And we need all of these competing groups that each want to mobilize their own product to somehow collaborate to let us figure out who's got the best one. In order to get these groups to want to collaborate, we have to protect their interests in some way. We have to protect their intellectual property and the investment they've made in this discovery pipeline. So how do you do all that, and how do you do it quickly? It turns out I've got some experience, because that's exactly what we did for Ebola virus. In 2013, which was right before the very large Ebola virus outbreak in Africa, this was the state of art for antibody therapy. On the left, we had an antibody. I'm showing it to you in orange. It's one arm of that Y. It looked fantastic in the lab. It could neutralize the virus, the best that we'd ever seen, but it didn't protect living things. They all died. And then we had this mixture of 3 antibodies, the blue and purple ones, that if you put them together, they would protect living things, but they didn't look like anything in the lab experiments by which we tried to figure out which ones would neutralize the virus. So we were at a bit of an impasse. We didn't really know what was going to be the right therapy. We thought we could do a whole lot better than the one on the right still, because it needed a massive dose. Maybe this data was telling us that we needed a combination of different antibodies working together. Maybe because these purple ones and blue ones, they worked better in one cell type than other. They worked a lot better. They gave 100% survival if you made them nicotiniana cells, and only half the animals survived if you made them in CHO cells. And that difference is the structure of the base that recruits immune help. Maybe we need immune help. The other thing is we might be doing the wrong kinds of experiments in the lab in cell culture dishes to predict what's going to protect a living thing. We, as a whole research field, were missing information. We needed better tools. We needed to look at more samples. And the trouble was that every one of these competing groups, whether it's an academic lab and a small company or a large company, they're all working in a silo. And the pandemic was threatening to escalate. We needed to very quickly find a way to amass a broader set of samples, to study, a broader set of expertise to understand what we were missing. We needed to evaluate these things side by side, and we weren't going to evaluate these things side by side in an apples-to-apples comparison of what antibody was best, unless everyone agreed to hand them all over to sit in the same room and look at them side by side in the same experiments. And we needed to figure out how to do all this very, very quickly. In short, we needed to be smarter, we needed to work faster, we needed to find ways to work together that we never had before. And so the question was, can we do all three of those things at the same time? Can we find ways, systems, frameworks, and legal documents by which we can get multiple competing interests to collaborate within the framework of what made them innovative in the first place? The competition in biomedical research gets results faster. Competition drives innovation. Competition improves biomedical research. Can we build a research network that allows the best of both of those to exist at the same time? We devised that framework by blinding people's samples to make the study very fair. Nobody knew who antibody was whose. We're just getting the data on what it was that worked and why. We protected their intellectual property. We ran parallel experimental tracks to put something in place as soon as possible and figure out something better later, kind of a tortoise and the hare strategy. And from that was born a study we called a VIC, the Viral Hemorrhagic Fever Immunotherapeutic Consortium. Ultimately, we united 43 previously competing partners across 5 continents to make a single study to figure out once and for all what's the right therapy against Ebola virus. So we united 100 people working together for 3 years, and these are the antibody samples that everybody contributed, and this is everything we figured out about them. And so we're able to figure out every feature about these antibodies, which things track together, which things track with saving lives of living things. Which of these features were most, that we could measure high throughput in cell dishes, would most likely predict protection in the animal model? And we could figure out if we did, the top 8 experiments with an area under the curve, 0.958, that's 96% accuracy, what was going to save the life of a living thing. So like I said, my father's daughter, we have an equation of how we can calculate and predict what's going to deliver protection. We saw that what the research field had looked at before was only moderately predictive. It's not very important. We found better ways of measuring neutralization, and we got them established in the research field faster because we'd done it with all the labs at the same time, working in everybody's samples. We found new things we never learned before about the ability of an antibody to bring about immune protection, the ability of that base to recruit immune cells to clear the virus. And all of that led to, finally, a treatment for a virus that was previously thought to be untreatable. This is the best therapy found against Ebola virus today. This is a cryo-electron microscopy structure from my laboratory. At the core of this structure is the viral spike of Ebola virus, and then the gray arms coming down from the top or in from the sides, the top and bottom, are 3 different human antibodies that inactivate it in different ways. We're able to show by doing this high resolution structural work how these antibodies defeat the virus. So the one at the top, the blue one, blocks the ability of the virus to attach to your cells. The light blue one recruits an immune cell health. The orange one stops the virus from being able to enter the cells. And we can look at a very precise molecular level of how these interact with the virus and how they work. And so that was the research effort I was leading when SARS-CoV-2 broke out 6 months ago. And the Gates Foundation called and asked if I would do the same thing for coronavirus. And so this is what we built, the Coronavirus Immunotherapy Consortium, where we are similarly uniting all of these competing interests and all of their different antibody therapies to analyze them side by side, an apples-to-apples comparison of which ones are really best, what makes them best, which ones remain responsive to different mutations that are emerging in the virus. How do we engineer these antibodies to make them even better than the first version we came up with? And we don't have 3 years to do this. We are instead undergoing a larger effort in a compressed amount of time. So what happens is that we now have all of these competing companies and academic labs that have these antibody therapies, and they are sending them here to La Jolla Institute for Immunology. We are blinding them in order to protect their intellectual property. We are sending these antibody samples out to experts in the field for everyone to analyze something different about how antibodies might work. We're cataloging this information in a database that's going to be publicly available to researchers around the world, figuring out from that which are the very best antibody therapies and making sure that these are mobilized wherever they are needed. There are benefits to the companies. When we look at their antibody therapy and we say, yes, that is the best, this is the one that should be launched for this population, that population, that data goes to the Gates Foundation and to the United States government to figure out what they want to invest in. They can also use this tremendous array of data that we are generating for their own IND filings. There's a benefit for them to contribute, otherwise they wouldn't contribute. But we're a nonprofit effort just aimed at making these therapies available for you and making sure they are well characterized. To understand how they work, we're undergoing lots of different kinds of complementary experiments to figure out every different way in which we understand what an antibody does so we can put together the right combination. And different groups are supporting different aspects of this study, including a private family that came in just at a time when we realized the coronavirus is mutated and we need to understand how to respond to that. Our first goals of this consortium are very much product driven. What are the best antibody therapies? We call them cocktails we put two together that we can deliver to people to save lives. Which ones can we mobilize anywhere in the world? But while we're at it, we're going to build more information than the world has ever had against antibodies against any coronavirus. We will understand what makes an effective therapy, what makes antibodies work together with greater synergy for more effective therapy. And this will help us respond to recurring seasons and recurring viruses in case, God forbid, there's SARS coronavirus 3. This will be publicly available information available to everyone. Our goal is to save lives, either by treating mild to moderate disease and preventing it from becoming severe disease, or to prevent people from getting sick in the first place, especially people that are highly vulnerable and people on the front lines. And this is what it looks like now. This is my program manager, Dr. Sharon Schindel, and the different antibody samples. It's been a lot of negotiation, a lot of phone calls, a lot of Zoom meetings, but it's now working and the data is rolling in. We've gotten all these different groups to collaborate. The way that my group in particular is analyzing this. We have the most powerful electron microscope commercially available. Here it is being built. It is not a 2-foot-tall microscope like you used in high school biology class to look at a frog. This is 11 feet tall, and it shoots a powerful electron beam, 300,000 kiloelectron volts down the center to illuminate very precisely the individual atoms and atomic bonds by which a virus becomes pathogenic and the immune system defeats it. Using microscopes like this, we can see very precisely how these therapies work, and we can engineer them to make them better. We can engineer the viral spike to make it a better immunogen. We can engineer the antibody to make it better therapy. So this is what an image looks like from this microscope. These are images of those viral spikes being attacked and inactivated by human antibody therapy. The molecules you are looking at are thousands of times smaller than the width of a human hair. These are so small that they were previously submicroscopic. It is only with this 11-foot-tall microscope and the associated equipment that we're able to get these insights. And that's what it looks like. We can understand how different immune molecules attack and where the sites are and how they work. For the coronavirus in particular, we are looking at different versions we are engineered of that surface spike. So here's 4 different snapshots in the range by which that thing moves and breathes. All biological molecules, just like biological people, move and adjust and breathe. And this thing undergoes small shape adjustments to try to evade itself for immune systems. And so by studying the forms and shapes that it adopts, and which of them we want to attack for immune defense, we can figure out how to better engineer snapshots of the molecule that I'm showing you here and to be a better vaccine or better diagnostic, and then better engineer those antibody therapies to work better next season. So these high resolution images that we make are the blueprints that help us make better therapeutics, better diagnostics, and a better vaccine. A lot of engineers make bridges and buildings. I'm a protein engineer. I make better molecules for better immune defense. So we're engineering that spike on the surface of the coronavirus to be better ordered and more stable and better present that right shape to get an immune response from everybody, to boost that immune response out. We want to inspire better quality antibodies, longer lasting antibodies, and we want to make better diagnostics to detect infection. And the goal is that by putting together that molecular engineering with our understanding that it gives us of human clinical immunology, how your immune system keeps you safe, not just from infectious disease, but cancer and autoimmunity, we can better design and deliver life-saving treatments. They're going to get us out of our houses, back to work, back to school, back with our loved ones. So right now, what we're doing in the lab behind me is that we're engineering spike to be a better vaccine and better diagnostic tool. We're discovering new antibodies against forms of that spike we've engineered, and we're understanding your human immune response. When it succeeds, when it fails, what we need to learn to better protect people. And there's a couple of things that we need to get there faster if you're in any position to help. We need an instrument to help us figure out better if we're making the right stable molecules. And we need to expand our clinical core, because those human donated samples, whether it's saliva to detect infection or blood to measure an immune response, we need more of them to understand if it works in all ages and all people and at all times. We need to expand our capacity of being able to collect and process those human samples in order to understand immunity. This is where we work, the Jolla Institute for Immunology. And we have three foci: infectious disease, cancer, and autoimmunity. You think of them as separate, but what we learn from one influences in others. So for example, basic molecules learned in studying autoimmunity provide a new therapy that damps down that overactive immune response and ultimate lung disease. We understand how viruses become triggers of cancer and triggers of autoimmune disease. When we find that viral trigger, we can learn how to design a vaccine to eliminate some of these diseases from human populations. Although we have three foci, it is one human immune system, and that one human immune system will find and destroy viruses and bacteria every single day. And your one human immune system will find and destroy individual cancer cells without you even knowing it, preventing them from turning into tumors. We are 21 individual labs, but we work together with a single focus to understand immune signaling and immune health. Thank you very much for your attention, and I'm happy to answer any questions. I have a couple of extra slides with some other information. Let me look at the questions.
Dr. Saphire, thanks again for all this information. This is a lot of depth into understanding deeper knowledge of COVID and all the different variables. So we really appreciate your time on this. We had a couple of questions. I think one of the common questions we received was really how dangerous is it to re-enter into the world again, from kids going back to school to traveling. What are your thoughts on some of that?
Yeah, I can put up some information I found. So I thought we might think about that. So first of all, I'm one of the 12 vaccine experts that predicts this clock. They average our responses together. I think I was more bullish and thought we were at 6 a.m., but we have a long way to go. There have been studies about how dangerous it is to travel. So for example, if you look at train travel within China during the peak of that outbreak, whether or not you became infected from a person sitting next to you had to do with how close you sat to them and how long you traveled together. So if you sat next to an infected person, there's an attack rate of 3.5%. So that means there's a 3.5% chance that you would be infected by them. If you're in the set that's sitting next to them, if you're in the same row of them, lower 1.5%, a couple rows away, very low. So, and going back to, so on the issue of can we travel, as long as the people sitting around you are not infected, it is perfectly safe. But how do you know if someone's been infected or not? And the answer is that it's a risk-benefit analysis. And then going back to school, there's a lot that we've learned. When we first studied children earlier in the outbreak, it's really clear that they don't get sick as much. That's good. But we now see that they replicate a lot of viral titer. We don't know if they shed, especially if they're asymptomatic. When we look at schools in Europe that have reopened, as long as the children were some distance apart and wearing masks, they found that there wasn't a lot of child-to-child transmission in the school. It wasn't the primary cause of cases in children. And that schools are equal or lower in transmission than other settings of equivalent density. So we have some data that suggests that there's some hope that we can go back, but we are not really safe, and we are not really without that risk until we have the right vaccines and the right therapies to protect people.
What about travels? Are you, you think that's besides the train, what about short flights?
Yeah, so on an airplane, I looked into this in Ebola, and Boeing and Airbus sent me their flight patterns. Air comes from the vents above you straight down, and there are HEPA filters on the floor. And so the air is constantly HEPA filtered. You are at risk really from the people in your area, the people in your row, and the people right in front and right behind you. And so it's a risk-benefit analysis. Do you need to travel or do you not need to travel? Are you immunologically vulnerable or are you less vulnerable? Is it a long flight? Is it a short flight? If the people sitting in your row are your own family, well, that limits your risk to people you only are exposed to anyway. And you can pick one of those airlines that doesn't sell middle seats to make sure that you've limited the number of people. So for everything, whether it's school or going out of your house, it's a risk-benefit analysis for how much you need to go and how vulnerable you are individually and immunologically.
We've got quite a few questions here, and I know we're running out of time. So, but I do have a question that I think is a good question is, what is the track record for RNA vaccines to date?
We have 20 approved vaccines. We typically have greater success developing a vaccine if infection has given you lifelong immunity anyway. So if you have had a measles infection, you tend to carry, or chickenpox infection is a great example, you carry those antibodies the rest of your life and you won't be reinfected. We have had a harder time developing vaccines in general against respiratory viruses for which you don't have lifelong immunity. So there are underlying mechanisms that we need to understand about how to inspire a memory response. And until we figure that out, we need to think about seasonal vaccines, like your seasonal flu vaccine, your seasonal coronavirus vaccine. Now, somebody asked, was something that deactivates the spike work for other coronaviruses? Absolutely. A lot of what we have begun with against SARS-CoV-2, we learned from studying SARS-CoV-1 and studying MERS and understanding the common colds. And we are sick of getting the common cold too. And so there's a lot we can do and learn from finding things that attack this coronavirus to apply to other coronaviruses. Someone else asked, what is the rationale that shot 1 antibodies are not neutralizing while shot 2 are neutralizing? You can actually test them. You can actually take the antibodies that the person has made at that time point, and you can do a lab experiment and say, do you neutralize the virus? And for the most part, the antibodies at that point in time did not. After the person had gotten the booster shot, those antibodies did. So the rationale are actual laboratory experiments, and we look at neutralization lots of different ways and lots of different cell types. They mostly weren't neutralizing early, they were neutralizing later. That booster proliferated the immune response and caused additional, what we call somatic hypermutation. Those antibodies evolved to be better and evolved to be neutralizing. Let's see, someone has asked, the economic and cultural impact of this virus has been unparalleled. There are a couple of things about this virus. One is that it is a respiratory virus, and these kinds of things spread easily. It's not like you can stop breathing. You know, Ebola doesn't spread so far because you get real sick real fast, and so you don't travel much if you have Ebola. And the other thing is that you contract Ebola by touching the blood, the diarrhea, or the corpse. And we are socially inclined to avoid blood, diarrhea, and corpses, but we are not inclined to avoid breathing. So a respiratory virus spreads among all of us. The other issue is that this particular respiratory virus sheds in people that don't yet know they're sick, the pre-symptomatic people. You can shed virus for 2 days before you know you're sick. And so even though we have been brought up by our mothers to stay home if we're sick and cover our sneezes, but if you don't know you're sick yet, you might not know to stay home, but yet you're shedding virus around you. And as a population, we coexist. We live in cities, we fly in planes, we go in and out of the taxi cab, we go in and out of the shared room and shared building. We occupy each other's space and objects and air. And that is why this spreads so quickly. It's also novel. Nobody has any immunity. We're all quite vulnerable.
So with that, Dr. Saphire, what is it that we can do to help? And what can we do to individually contribute to help the situation?
I could use some help at better analyzing those human clinical samples, because all the lab experiments in the world are only as valid as your ability to apply them to real people in the variety of genders and ages and populations. And we need to receive and process and study more of those human samples from healthy people, from asymptomatic people, from vaccinated people. So we could use some help getting the equipment and the personnel we need to expand those studies to get them done faster. We need those results right away. We can't wait a year.
Right. Wonderful. Well, thank you again, Dr. Saphire. And to be respectful of your time and everyone else on the call, we'd like to conclude the call today. But again, thank you, La Jolla Institute of Immunology and Dr. Saphire. And, Dr. Saphire offered up, if anyone has questions offline, she'd be more than happy to chat with any of our Beta Wealth clients on any questions that we weren't able to get to today. So again, thank you very, very much. And I hope everyone has a great week. Thank you.